So hi everyone again. I am Manoel Neres. Thank you for this opportunity. And now I will present you my postdoctoral project. Uh, this work is called the Host-pathogen interactions and immunomodulations of Mycoplasma mycoids synthetic genome. This work is supervised by Professor Dr. Lucas Miranda Marques from the Federal University of Bahia, Brazil. Well, during this work, we will go over some topics. The first one, a brief introductions, main goals, methodology, followed by future directions. So, molecular classes comprise about 200 species, widely found in plants, animals, and humans. Mycoplasma, ureaplasma, bacterioplasma are known to inhabit animals as commensal, saprophyte, and pathogen. So these microorganisms can colonize many type of growths. For some years, we have been working on host-pathogen interactions and immunomodulations of molecules. And now we are interested in these interactions between mycoplasma, mycoids, and your animal growth. So mycoplasma mycoids clusters is now by causing diseases in men, ruminants, cattle, goats, and sheep. With this, the objective of this study is evaluating the microorganism growth interactions and immunomodulations of streams with synthetic genome of Mycoplasma mycoids in cell culture and good animal model. So for this, we divided our methodology in 30 stages. First one, Mycoplasma mycoids growth, and first go to good collections. We start this step as soon as you're receiving the strands, the genomic synthetic strands, approximately 18 days ago. So you already done this stage. The first stage in the determinations of cultivation time and inoculation bacterial concentrations. This stage is already done too, and it's a recent data, but we can see that high concentrations of mycoplasma and inhibit blood cell of goats growth. So the next two stages are cell culture model and animal model. For cell culture model, goat blood will be collected and the PBMC will be separated by fecal gradient. After PBMC extractions, the cell culture will be affected with minimizing genome strand and wide strand. How you can see here, all this minimizing genome strand and a wide strand. So after the periods of inoculations, we were having the cell and a supernative. In the cell fractions, we will be realizing some dosages and qPCR. A qPCR in a qPCR we will analyze inducible nitric oxide synthesis, and by flow cytometry we will verify immune proliferations, apoptosis assay, and cell suppression marker. In the supernatant fractions, we will verify nitrogen oxide and oxygen peroxide, and those agents in immunological parameters like all these cytokines by ELISA test. In the animal model, will be divided into 6 groups, 4 groups for inoculations with synthetic genome Mycoplasma mycoids, 1 group inoculated with wide stream. In a 6th group, there is animal not affected. So Mycoplasma mycoids will be inoculated via trachea. This animal will be evaluated for a period of 60 days with blood collections at interval of 15 days. For 60 days, mycoplasma detections and quantifications will be performed using real-time PCR and filter. At this time, histological and immunohistochemical analysis sections of lung tissue will be used for the identifications of these cytokines. Well, future directions we have done these 2 stages, and for future directions we have to make a cell culture model and an animal model. So thank you, more time, for this opportunity.
Thank you. Questions? Anyone? One thing I'm wondering. Go ahead. I heard a voice.
I'm Lucas. We just finished the Alamar Blue test. Now we made the experiment this week. We will analyze carefully the data to use the better dose to try and to make the culture cell methods. We are defining now the best dose to understand how the white stripes and synthetic strands are in the I'm sorry, I'm nervous, and my English is not better when I'm nervous. We are defining the better dose to try to make the other experiments. We need to understand how the strands behaviors in the macrophage culture. And when we finish this data, we will started another experiment with the cytokine dosage, with the flow cytometry mark cells.
Lucas, how many mycoplasma organisms do you plan to inoculate each goat with? I know that there was a group in Kenya that previously did similar experiments with a partially minimized cell. What are you following their protocols or something different?
We will make the same similar protocols with 4 synthetic mycoplasmas and with wild strain, but we will analyze more. We will make more analysis because we will use the flow cytometry to cells, and we will use immunohistochemistry to the tissue, and the Kenya group don't realize this experiment in the cells.
Okay. And so when do you plan to inoculate goats?
We need to finish the cell model first to determine how how we can do and with which strands we will use for the animal model. You finish the cell model first to decide with the which strand we will use for the animal model.
Okay, cool.